So in this presentation, my purpose is to give a brief overview of how can we use microbiome research to answer questions related to improving animal performance and health in swine systems, but these methodologies can actually be applied for any other food animal system as well. So in this presentation, I will give some information that most of the audience probably already are familiar with, but I would like to know, or I would like to have or present aspects that we can take or pay attention to as far as using the microbiome or harnessing the power of microbiome science to answer questions related to swine health and nutrition. So this is a real picture of a fecal sample and microbial communities. Of course, the picture has been artistically enhanced, but it depicts some of the aspects that I consider most fascinating about microbiome science today, and the power of DNA sequencing to answer questions about the influence of the microbiome in performance and health. And it's the fact that the microbiome in different systems, particularly in the gut, this is a fecal sample, is an incredibly diverse ecosystem. There are complex interactions between different kinds of bacteria, and there's also interactions between bacteria, feeding products, as well as between bacteria, feed, and the host. So, at the same time, this is one of the most challenging aspects of studying microbiomes today. Being able to recapitulate this complexity and manipulate these microbiomes for purposes of improving performance and health in animal systems. The gut microbiome, and this is something that most of the audience already knows, is vital and critical in nutrient processing and immune regulation. So there are complex carbohydrate substrates that mammals cannot degrade because we lack the enzymes to degrade them. So these complex carbohydrates pass on digested through the upper GI, and they concentrate in the colon, where a very specific group of bacteria can break them down and ferment them into short-chain fatty acids. These short-chain fatty acids are mainly acetate, propionate, and butyrate. Acetate and propionate are absorbed by colonocytes, and they travel through portal blood to skeletal muscle, where they constitute a significant source of energy. Butyrate is the main fuel source for colonocytes and also plays a critical role in maintaining intestinal barrier integrity. At the same time, we know that there's a specific and, let's say, homeostatic community of microbes in the gut ecosystem, which can be disrupted with different stimuli from the outside, namely diet, antibiotics, or stress. When this equilibrium is broken, the immune system is able to sense it, and they can be a trigger of pro or anti-inflammatory reactions in the colonic system or in the gut system as well. This, we believe, is the principle behind many of the diseases that we see today, especially autoimmune disease in humans, right? The fact that our immune system is able to coexist or to live in a homeostatic relationship with the microbiome, and the fact that any changes in the microbiome, namely by these external sources, can trigger pro or anti-inflammatory reactions as well. So, studying the microbiome in swine performance and health is very challenging. We believe that maybe diet, nutrients and feed additives are the main modulators of the gut microbiome, but there's also stress, at the time of weaning, in the form of disease and heat. There's also microbial communities in the respiratory tract and the reproductive tract that interact with the gut microbiome as well by molecular cross-fitting. But there's also an environment that constantly seeds microbes through the animal from outside. So it is very challenging to see how we can manipulate microbial communities in the gut given all this complexity as well. So in theory, what we want is being able to manipulate the microbiome through diet and environmental manipulation to either boost the abundance of microbes of interest or eliminate undesirable microbes, especially those that cause disease. At the end of the day, what we want is to manipulate the microbiome to increase performance and health and target specific desired phenotypes. in theory, manipulating the microbiome is a more efficient and less time-consuming manner of improving phenotype than genetic improvement. So that's one of the promises of the microbiome today. However, there's a lot of questions surrounding the fact of what is the real quality of information that I can obtain from microbiomes? How can we use microbiome power to manipulate animals in the way we want? So, gladly, we're in the time right now of big data and genomics. So right now it's very easier than 50 years ago when we rely on culture-dependent techniques to profile the DNA of hundreds and thousands of samples simultaneously. Sequence the DNA and have very fine, detailed resolution of the kind of microbes that live in a given microbial ecosystem and the kind of functions that they can potentially perform. This is the advantage of microbiome science in the big data and genomic era today. There's also specific bioinformatic tools that allow us to mine this data and look for specific patterns. That is, identifying microbes that can either characterize animals that are healthy from animals that are diseased, animals that are lagging behind in growth from animals that are more efficient as far as growth is concerned. Also, the microbiome is a very powerful tool for prediction and diagnosis. Looking at microbial communities, we're able to predict or diagnose given phenotypes in different animal systems. So if I study the microbiome, as Dr. Costa pointed out before, I can potentially able to differentiate animals that are healthy from animals that are diseased. And this is something that the microbiome science provides today that we weren't able to do, let's say, 50 years ago. But to start harnessing the power of microbiomes in our advantage, we need to start surveying the microbiome from a compositional perspective in different systems. In this study, authors took data from 20 publicly available data sets from sequencing the 16S ribosomal RNA gene. The 16S ribosomal RNA gene is the gold standard for taxonomic studies in microbiome today. It answers the question of what kind of bacteria are there. It doesn't tell us anything about function. It just tells us in this microbial ecosystem we find A, B, C, D types of bacteria. Not surprisingly, these authors found that the microbial community composition in the distal gut is significantly different between that found in the upper GI. This is what we call an ordination technique or an abstract map. It's what microbial ecologists is the main tool used by microbial ecologists to interrogate microbial communities. So every single point or dot represents the microbiome of a single pig or a single animal. The closer the points are together, the more similar the microbiomes are. And the farther apart they're from each other is because the microbiomes are more different. So we see that there is significant discrimination between the microbiome found in the distal gut from that found in the upper GI, which is not surprising based on the theory of the environment selects. Different physical chemical conditions along different segments of the GI select for the type of microbes that you're going to find along. Another interesting aspect of this research is that authors were able to identify a core microbiome. This idea of a core microbiome has, received increasing attention since the microbiome boom, let's say 10 or 15 years ago. What is the set core of bacteria that all, let's say, humans possess? These authors found that in the 20 publicly available data sets, we could identify a specific group of bacteria that are present in 99% of the pigs studied. Not surprisingly, these bacteria are all associated with the breakdown and metabolism of complex carbohydrates. For example, Ruminococcus, Blautia, Prevotella. Those are taxa that are typically associated with the breakdown and fermentation of fiber. So I think it's interesting to see that the core microbiome of swine is mainly composed of bacteria that have the capability to degrade complex carbohydrates. So an interesting question is, for the purpose of manipulating the microbiome, should we concentrate in these core microbial communities, or should we concentrate in communities that are more transient, or that belong to only specific groups of pigs or individuals per se? Another interesting fact about the microbiome and along the different segments of the GI in the pig is that the distal gut has way more diversity than the upper GI. Mainly, there's more types and different kinds of bacteria in the colon that we find in the duodenum, jejunum, and ileum. This is interesting because we still don't know. There are no clear studies looking at the function of the microbiome in more proximal sections of the GI. We don't know what the function is exactly of the microbiome that populates the duodenum, the jejunum, and ileum. There has been more work done in the proximal and distal gut. I'm sorry, in the distal colon. And it's not surprising because the distal colon and the cecum is the main fermentation chamber. There's also studies that consider dynamics of the microbiome from farrow to finish. Here you can see another ordination plot showing microbial communities distinctions at different ages in the duodenum and ileum. You can see some segmentation. You can see that the samples taken at 0 and 10 days of age and 21 of age discriminate themselves for those samples taken at market ages. But these distinctions are even more dramatic and more extreme in the distal gut, in the cecum and the colon. Does it mean that is it easier to manipulate the distal gut microbiome than the microbiome that we find in upper sections of the GI? Diversity of the microbiome is also dynamic according to size and according to age. For instance, in the duodenum and jejunum, there seems to be a pattern of increased diversity as pigs age. That pattern is probably more clear in the duodenum and jejunum. But interestingly, in the distal gut, this pattern is even more clear, right? We see that there is a peak in diversity in the numbers and types of different microbes found at day 33, 62, and 84. Does it mean that if we plan for interventions that modulate the microbiome, should we concentrate in early stages of development rather than day 33, day 62? To answer this question, some researchers have even implemented environmental interventions, let's say, to boost maturation of the gut microbiome. For instance, in this work, they raise piglets in either conventional conditions or adding topsoil to the environment. They saw that when they added topsoil to the environment, the piglets at day 13 harbor more or significantly more abundance of taxa typically associated with the degradation of complex carbohydrates, particularly Prevotella, which is one of the members of the core microbiome. I think this is very interesting because it means that by providing topsoil to rearing conditions, we're accelerating maturation of the microbiome in a way pigs acquire this enzymatic machinery to break down complex carbohydrates in a more efficient way. This boost on the microbial communities capable of degrading complex carbohydrates extend into weaning, with the abundances of Prevotella more significantly abundant in pigs rearing topsoil versus conventional. And they even detected that potential pathogens such as Campylobacter or the sulfovibrio were diminished in abundance at this age in pigs that were reared in topsoil conditions. So these hypothesis-generating microbiome experiments, which we unfairly design sometimes as fishing expeditions, are very important. Because they offer questions, they offer open questions that we can target to start considering manipulating the microbiome in our favor. For instance, do we focus on the core or the transient microbiome to boost health and performance in piglets? There are wide biogeographical differences, across all the segments of the GI. What side is more important? Is it the upper GI, the ileum, duodenum, or is the distal gut? What is the most critical developmental stage to target interventions? Is it the early ages, pre-weaning, or is the late stages before market? And more importantly, These previous studies that I mentioned only offer a view on composition of the microbiome. Who is there? But they didn't tell us anything about what microbes are doing. In that regard, I think it's very important to start using functional rather than compositional surveys on the microbiome. The compositional surveys of the microbiome are mainly based on the sequencing of this gene. It's the 16S ribosomal RNA gene, which is the taxonomy marker most frequently used in microbial ecology studies. This is approximately a 1.5 kilobases gene, which is composed by very variable regions alternated with some conserved regions. The secret is that we anneal primers, or we use primers that anneal to the conserved regions so we can amplify these variable regions. As you can see, there are 9 different variable regions in the 16S gene. However, current sequencing methodologies today, at least the ones that are more cost-effective, doesn't allow us to sequence the whole 16S, which would increase taxonomic resolution. We can only sequence portion of it, either targeting the V1, V2 region, or the V3, V4, or only the V4, right? Here, you can see that there's significant differences in microbiome studies by using different regions of the 16S. So these are the same samples, sequenced either using the V1 to V3, the V3 region, V3 to V4, or only V4. So imagine if you wanted to do a meta-analysis. A meta-analysis would be futile if you have different segments of the 16S sequence as opposed to the whole 16S. In fact, we demonstrated that by using different regions in the 16S gene, we can obtain different information. For instance, this is the full length this is the taxonomic composition based on the full length sequencing of the 16S gene compared to only sequencing the V2, the V4, or the V6 regions. As you can see, the V6 region overestimates diversity by amplifying supposedly taxa that is not present in the ecosystem. Probably the region that is most closely resembling the whole length 16S genes, and is the region that we most use today in microbial ecology studies, is the V4. The V4 is the one that most closely resembles the full length gene. It's important to say that most of the studies that we have today in microbial ecology use this compositional surveys. Very few studies are using metagenomics, which is a technique that accounts for all the different genes potentially harboring that microbial ecosystem. So it's the same technique. We sequence, there's a mix of bacterial genomes in any environmental sample, and we use shotgun metagenomics to tease apart or to sequence specific segments randomly, specific segments of different functions found in that ecosystem. The problem is that shotgun metagenomic sequence is orders of magnitude more expensive than the 16S procedure, probably 3 to 4 times, depending on how deep you go into sequencing. However, there are some studies that have used shotgun metagenomics to profile, what is the functional potential of the microbiome of different stages of development. Not surprisingly, in nursing, you have, in the microbiome, you have more abundant of proteins associated with lactose utilization. And then after weaning, the abundance of genes that are associated with the degradation of complex carbohydrates or plant-derived sources is more abundant. However, these authors were only able to detect significant differences only in a few genes, just because they only took 3 or 4 samples for metagenomics due to price or budget constraints. There's another study in which they study the metagenome of high efficiency or pigs that are more efficient in terms of feed efficiency and pigs that are not that efficient. And they found that, for example, phosphotransferase systems, PTS systems, are more abundant in pigs that have high feed efficiency. This is interesting considering the phosphotransferase systems, or PTS, we have it seeded as enriched in, for example, human studies in human groups that rely on more digestible carbohydrate sources, as opposed to using more fiber. Does it mean that using more digestible carbohydrate sources would yield pigs that are more efficient as far as feeding conversions? Another important aspect of studying the microbiome in swine systems is that the microbiome is only one layer of information of everything else that happens in the animal. What happens with GI physiology, for instance? What happens with the metabolites produced that Dr. Costa mentioned before? What happens with functional genomics? And what happens with nutrients? How can we integrate all this information to have a better perspective of how the microbiome impacts performance and health in the pig? I think this is a very important direction that the field is taking right now. Integrating different types of data sets to have better predictions based in microbiome composition and function in different studies. However, these are very costly experiments, mainly because the cost of metagenomics, metabolomics techniques are still cost prohibitive in some cases, and also because if you add the cost of these kind of sequencing techniques to the cost of rearing and keeping animals, that amounts, or escalates really quickly as far as what you can achieve in this experiment. There's very few studies that actually profile, for example, or correlate microbial community composition with gene expression along the GI. In this study, for instance, which is not based on high-throughput sequencing techniques, but only on microarrays, they show that, for example, in the jejunum, there's some microbes that significantly correlate with gene expression. Every single blue symbol here corresponds to a gene along the jejunum, and the purple dots indicate or show specific bacterial taxa. They showed that, for example, in the jejunum, Ruminococcus, Faecalibacterium, Eubacterium, and Butyrvibrio are significantly possibly correlated or negatively correlated with the expression of some genes in the jejunum. But I find this a little strange because Ruminococcus, Faecalibacterium, and Butyrvibrio are not bacteria that are typically found in that GI segment. So this is the limitation of this kind of study, especially when you don't use high-throughput sequencing techniques. Lately, we're trying to develop or standardize these kind of techniques at the University of Minnesota in our integrated animal system biology team, in which we try to dissect the animal into different segments using high-throughput omic techniques to have a better understanding of how nutrients, the host, microbes interact to impact pig performance. There's also the need today of implementing mechanistic models to understand the structure and function of the swine gut microbiome. In the human microbiome field, we have relied on no-biotic mice most of the time to being able to manipulate and to transfer microbes from one, let's say, diseased mice to a mice that doesn't show the disease symptoms and see if we can recapitulate or reproduce the same condition. In pigs, there's the promise of using these no-biotic mini-pigs models, but these are extremely expensive. I mean, ideally, every single microbiome lab would have this kind of mechanistic models to understand how the microbiome can be manipulated to influence physiology. But these methods are still very cost prohibitive. There are some techniques that have been developed in the human microbiome field, for example, the SHIME, which is a series of chambers that simulate fermentation and physiological dynamics across different sections of the GI, in the stomach, in the small intestine, ascending colon, etc. So it's a question of manipulating pH using different substrates of interest and see how the microbiome transfer from a fecal sample reacts to those different conditions. There's other models, for example, the TEAM model, which is even way more complex. And it also has the same purpose, manipulating microbiomes in different chambers in which you also control the pH and different physiological conditions to be able to see how microbes react to certain stimuli. The main problem with these tools is that these tools don't have any cells, any host cells, so that you can also see or study interactions between the microbiome and the host and feed. They're just, the only thing that you can determine from these models is whether changing different physical-chemical conditions will change also the microbial communities. But there is no information about the host. There are some models that includes gut epithelia and mini chambers in which can inoculate with different substrates as well and different microbes just to see what happens in the gut epithelium. The main problem with this method, especially this gut-on-a-chip system, is that it's very restrictive as far as the microbes that you can manipulate. Usually manipulated anaerobic or strictly anaerobic microbes, which are the main dwellers of the distal colon, is very difficult or almost impossible. Now, I think one of the things that have better promise today as far as manipulating the microbiome in vitro systems is enteroids. So Dr. Saki Salz's part of our integrated system biology group at the University of Minnesota is developing some swine enteroids. And she has been having success into using the swine enteroids, which are three-dimensional structures that have all the cells and all the types of different cells that compose the small intestine, just to see how inoculating those enteroids can trigger or can change the dynamics of GI physiology. And there's going to be, our next speaker is going to talk about this system more in detail, but I think this is one of the most promising areas of using in vitro models to understand the microbiome. Right now, she has been having success in using feed additives, challenging with different pathogens, and analyzing gene expression patterns in enteroids. But the purpose that we have in the future is to use whole microbiomes, to inoculate the enteroid with whole microbiomes just to see what happens as far as gene expression in the small intestine. And the idea would also be to use step-by-step feed additives, whole microbiomes, and also incorporating specific pathogens to see how the gut epithelium reacts. Okay, so in this talk, as you could see, I just presented an overview of the state-of-the-art of microbiome science today, in swine systems. And my purpose is basically to provide a series of steps that I consider are essential into advancing microbiome science in animal production systems today. As I said before, it is imperative that we keep funding more hypothesis-generating research in swine systems, right? We just need to know what happens, for example, in the early microbiome dynamics. What are the forces that seed the early microbiome in piglets, right? Especially across critical developmental stages, pre-weaning and post-weaning. We need to see what happens in these stages when we add, for example, specific feed additives and alternatives to antibiotics, just to see what kind of microbes we can recapitulate, what kind of microbes are boosted, what kind of microbes are diminished, and see whether those microbes are the potential probiotics of tomorrow. There is also the need of more functional microbiome surveys. Apart from the traditional 16S approach, which just answers the question, who's there? We need to know what microbes are doing. And for those methodologies, metagenomics is a very powerful tool. Also metabolomics that Dr. Costa talked about previously. And more recently, we have been using the concept of metatranscriptomics, sequencing the RNA of microbes in a given ecosystem. Also, we need to view animals as a highly integrated system using meta-omic data sets. Remember that the microbiome is just one layer in the animal, right? We need to know what happens with the metabolites that are produced. We need to know what kind of genes are overexpressed, given the presence of specific bacterial profiles, right? And to do that, we just need to integrate different types of data sets or different types of measurement from the same animal system. And last, it's very important to start using these mechanistic models, to understand how specific microbes and feed impact the host. And if you look at this seems to be a sequential series of steps for microbiome studies in swine systems, right? Everything begins with a hypothesis-generating technique. After you have identified different microbial community composition profiles that are different, then you want to know what are the functions of these microbial communities. Then you can see if you can get more information from other parts, other layers of the animal, whether it's gene expression or metabolomics. And then you want to recapitulate or you want to test those hypotheses in a system that is more mechanistic. So this was some of the things that we're doing in our lab right now. And I think this offers a series of steps for practices of microbiome research today that would benefit the industry and the field.